Hi, it's Miss Vitale. This unit is on genetics and it is created for AP Biology at Summit High School. Genetics is the scientific study of heredity. A diploid cell, as you know, has 2 copies of every gene. A cell that has 2 copies of each chromosome also has 2 copies of each gene. So that is the key to understanding genetics in higher organisms. Each copy of a gene may differ slightly from its counterpart on the matching chromosome. Alleles are alternative versions of the same gene. For example, there are different versions of the gene for eye color. There may be a blue one, there may be a white one. In this example, there are 2 different colors, purple and white, which are 2 different alleles for the flower color gene. Most genes are simply instructions for the production of a protein. Alleles are variations in those instructions. A trait like eye color, skin color, height, is a characteristic that is passed from parents to offspring. An organism is homozygous when it has 2 copies of the same allele. For example, if they have 2 blue eyes or 2 brown eyes. Heterozygous is when an organism has 2 different alleles for a single gene. For example, they may have 1 blue-eyed allele and 1 brown-eyed allele. In that case, if brown is dominant, they would have brown eyes. Here we show crosses between 2 parents. If you have one parent who has 2 dominant alleles, capital R, and the other parent who has a dominant and a recessive allele, capital R, lowercase r, they could produce offspring that are capital R, or capital R, lowercase r. Genotype is the genes that are present in a particular organism or cell, and we use the letters for dominant and recessive to indicate the genotype. Phenotype is the physical and or physiological traits of an organism. Can be considered the characteristic that is expressed by the genes. Gregor Mendel was an Austrian monk. He had a background in math, physics, and biology. He used pea plants in his studies because pea plants have flowers with an enclosed structure. That means that they self-fertilize. So by using a plant that had self-fertilizing enclosed flower structures, he could prevent the plants from cross-fertilizing. The only way that they could do that is if he physically took like a tiny little brush and transferred the pollen from one to another. So the enclosed flower structure also means that they self-fertilize. In addition, pea plants reproduce very quickly. It goes from seed to seed. In other words, when you plant a seed, it only takes 90 days for it to grow into an adult plant that is producing its own seeds. It also produces a large number of seeds. Another reason why pea plants were a great organism for his genetic studies is because, for the most part, they have 2 distinct forms of each trait. For example, seed shape is either round or wrinkled. Seed color is either yellow or green. Flower color is either purple or white. Pod color is either green or yellow. The pod shape is either smooth or wrinkled. Flower position is either axial, which means the flowers come off of the sides of the stems, or terminal, where the flowers are at the end of the stem. And plant height, where the plants were either tall or short. For the most part, these were the only 2 traits that appeared. You didn't really have a bunch of different forms of one trait, and by having only 2 distinct forms of each trait, it made it a lot easier for him to come to his conclusions. Mendel started his experiment with purebreds, or they may also be called truebreds. A purebred only produces one form of the trait, and a purebred comes from a line of all purebreds. So a purple flower that's a purebred comes from a line of all purple flowered plants. The white flower comes from a line of all white flowered plants. So he began with purebred purple and purebred white, and he cross-fertilized them. Again, I said he had to do that by hand. When he cross-fertilized them, the next generation, which is called the first filial generation, were all purple. However, these are not purebreds because their parents showed 2 different forms of the trait. Since they had 2 different parents, the F1 purples were considered hybrids. A hybrid, therefore, is the offspring of a cross between purebreds. Then he let those F1 purple hybrids self-fertilize, and in the second filial generation, 75% of the offspring were purple and a quarter were white. Mendel had to make sense of all of this data without the knowledge of genes and chromosomes. Remember, in the 1800s, there was not the knowledge of genes or chromosomes. He considered the meaning of the ratios and the phenotypes in his breeding experiments, and not only with flowers, but all of those other traits that I mentioned before. He would do the same thing, start with 2 purebreds for different alleles. He would cross them, and in the F2, he always got 75%, 25%. So his first conclusion was the law, which at the time was the principle, of segregation. And basically what that states is that each sexually reproducing organism has 2 determinants or factors. That's what he called them, because again, he didn't have knowledge of genes. Now we know that those factors were genes. So they have 2 factors for each characteristic. So that would be your 2 alleles for each characteristic. He also said that those factors separate or segregate during meiosis when gametes are produced. So in other words, the 2 alleles that are carried for one trait separate when gametes are being created. His second conclusion was what the law of independent assortment. This states that each pair of factors, or alleles as we now know, is distributed independently during the formation of gametes. In other words, it's a random assortment of those individual gametes, because one allele goes into half of the gametes, the other allele goes into the other half of the gametes, but which one is random? This basically means that the independent assortment of the homologous chromosomes during meiosis results in genetic combination. We've talked about that before. This is one of the things that contributes to the variation in sexually reproducing organisms. Probability is the likelihood of an event or outcome, the chance of something happening. Probabilities can be expressed as a fraction, a ratio, or a percentage, and the probability scale ranges from 0 to 1. 0 means the event is never going to happen, it's certain not to occur, and 1 means that the event is always going to happen and that it is certain to occur. There are rules for probability, and the first one is the rule of independent events. The outcome of an event is unaffected by previous events. If a couple has 4 boys, what is the probability that their 5th child is going to be a boy? It's 50-50. Because each child has a 50% chance of being a boy and a 50% chance of being a girl, and the previous children that you have do not affect the future children's probability of being a boy or a girl. The rule of multiplication is the probability of 2 or more events occurring together, and that is equal to the product of each individual's events probabilities. For example, the chance of you getting a head on a penny is a half. The chance of you getting a head on a nickel is a half. The chance of you flipping a penny and a nickel at the same time is a quarter, because you multiply the probability of penny heads, which is half, by the probability of nickel heads, which is half, and that equals 1/4. The rule of addition is the probability of an event that can occur in 2 or more different ways is the sum of those separate probability of those ways. In other words, if you add up all of the different probabilities of an event, it's going to add up to 1. The chance of you flipping a coin and getting heads is half. The chance of you getting tails is half. What is the chance of you getting heads or tails? That's 1/2 plus 1/2, which is equal to 1. Another example would be rolling a dice. The probability that you're going to get a 1 is 1/6. Probability that you're going to get a 2 is also 1/6. What is the probability that you're going to get a 1 or a 2? You add the 1/6 for the 1, the 1/6 for the 2, and you get 2/6, which is 1/3. The rule of multiple events states that the more times an event occurs, the closer the sum of these events will get to the expected probability. In other words, if you flip a coin 10 times, you may get 6 heads and 4 tails. If you flip a coin 1,000,000 times, you're more likely to get closer to the expected probability, which is 50-50. Pleiotrophy is when a single allele has more than one distinguishable phenotypic effect. The allele is then considered pleiotrophic. A good example is the allele that's responsible for the coloration of Siamese cats. Siamese cats have a light-colored body and dark tips or extremities. They also characteristically have crossed eyes. Both of these result from the same protein produced under the influence of the allele. So two different traits, crossed eyes and dark tips, are created by one allele. Another example is sickle cell disease. Sickle cell is caused by one defective allele, one recessive allele, and it causes misshaped red blood cells. Those misshaped red blood cells can block the circulatory system. They don't carry oxygen nearly as well, and sickle cell causes many symptoms. Weakness, anemia, can cause brain damage, heart damage, etc. So in other words, that one defective allele actually causes multiple different symptoms or multiple different phenotypes. Epistasis is when the phenotypic expression of one gene is affected by another gene. Our example here is mouse color. There are several genes that determined the coat color in mice. The wild color or the common color is called agouti. This is gray, and the reason that they appear gray is because they have bands on the individual hair of black and white. So a capital B determines that the hair is going to have bands and it's gray. So you can have capital B lowercase b or capital B and your hair is going to have bands. But if you have two lowercase b's, then the hair is unbanded and appears black. There's a second gene that determines an earlier step in the formation of hair color. The dominant, which we're using an A here, is for normal color development. So if you're capital A or capital A lowercase a, you're going to have normal color development. If you have two lowercase a's, it blocks the color development. So if you have two lowercase a's, you're going to be an albino regardless of whether you have a dominant B or not. So one gene is affecting the expression of another. Polygenic inheritance we've talked about a lot in class. These are characteristics that are controlled by groups of several genes called polygenes. Many of your physical characteristics are polygenic. Each allele for the trait intensifies or diminishes the observed phenotype. So as a result, there's variation in such characters is continuous or it's quantitative. In other words, you get a broad range of different phenotypes, like height. Okay, people come in all different heights. Skin color, eye color, hair color. There's a broad range of a bunch of different colors, and not only different colors, but there's variation in those colors. Okay, your eyes can be dark blue or light blue, and a bunch of different possibilities in between. Humans differ in the amount of pigment they have in the skin, which is what gives our skin different colors. The pigment that creates our skin color is called melanin. The amount of melanin in our skin cells is controlled by a lot of different genes. In addition, skin color is also controlled by the sun, because exposure to the sun increases melanin production. That's a really good example of how genotypes are affected by the environment. Oftentimes, our phenotype has more to do with environmental effects than with genetic effects. And recent studies show that the environment plays a bigger role than we previously thought in the expression of our genes. The Siamese cat that we talked about has a genotype that results in dark fur, but only at temperatures below normal body temperature. So the fur on its body is light and on its extremities is dark. Penetrance is the proportion of individuals in a group with a given genotype that actually shows the expected phenotype. People can have genotypes and it may not be expressed. For example, there are a lot of genes that predispose us to different types of cancer. Some people that carry those genes that predispose them to a cancer may never get that cancer. So penetrance is the amount of people that carry that gene that actually get cancer. Norm of reaction is the phenotypic range of a genotype. And again, there's going to be much larger range for polygenic traits because they are multifactorial. Multifactorial traits are influenced by a lot of different factors, as we mentioned before, genes and the environment. The expressivity of a genotype is the amount that it's expressed or the degree to which it is expressed. Think about how Siamese cats that are kept indoors would be different than Siamese cats that were kept outdoors. Since the color of their fur is influenced by temperature, the tips of the tail, the nose, the feet are cooler, lower body temperatures than their core. So cooler temperatures create the fur to be darker. Theoretically, if you strapped an ice cube or an ice pack on the back of a Siamese cat, over time you would develop a dark patch of fur on its body. One of the ways that we study a lot of genotype and phenotypes is identical twin studies, because identical twins are genetically identical. However, any differences between them can be 100% attributed to their environment. There are a lot of genetic disorders. Most of them are recessive disorders. Recessive disorders are inherited through recessive alleles. An allele that causes a genetic disorder codes for the wrong protein or no protein at all. Some of those proteins could be things like enzymes, receptor proteins, transport proteins, structural proteins, and the lack of that protein, whether it's defective or is never created, causes a set of symptoms. One of the reasons that recessive disorders are more common is because of carriers. Carriers are the heterozygote. A lot of times people don't know they're carriers because they have the dominant allele, they're normal, and they don't realize that they're carrying the recessive allele. But if two carriers have a child together, there is a 25% chance that that child is going to have that disorder. Some examples of recessive diseases are hemophilia. Hemophilia is when the blood cannot clot due to the lack of a certain protein. It is a recessive gene that is found on the X chromosome. Therefore, it only affects males. We'll talk about those types of genetic disorders later. So rarely women have this. And the lack of clotting doesn't just cause excessive bleeding, but it can cause the buildup of fluid and blood in your tissues. One of the tragic stories about hemophilia is that people who were hemophiliacs used to get clotting proteins from blood donations. People would donate blood, the clotting factors would be removed, and they would be put into a serum that would be injected into hemophiliacs, and that would be a type of treatment that they were taking. Unfortunately, in the late 70s and early 1980s, AIDS was in the blood supply and we didn't know it. A lot of hemophiliacs contracted AIDS because of the clotting factors that they were taken from donated blood. Now, clotting factors are reproduced in a lab using genetic engineering and bacteria. We'll also talk about that in the next unit. So that is considered a much safer way to provide hemophiliacs with the clotting factor. Sickle cell disease, which is found in the African American or black population, and about 1 out of every 400 have this disease, is when the red blood cells have an abnormal sickle or crescent shape. They tend to clump and block blood vessels. They can't carry oxygen. They cause weakness and pain. This sickle cell disease is due to the wrong protein. It's actually a substitution of a single amino acid in the hemoglobin protein. And the treatment for it is drugs and blood transfusions. Sickle cell is unique in that the heterozygote, the carrier, actually can have minor symptoms because they have a very small amount of sickle cells. That's about 1 out of every 100 African Americans. This is one of the reasons why people with sickle cell or people who are carriers are resistant to malaria. If you have any sickle-shaped blood cells, malaria doesn't like you. Malaria is a protist that lays its eggs in your red blood cells. Because the parasite spends part of its life cycle in the red blood cells, it doesn't like sickle cell. So people who have sickle cell or are carriers don't usually contract malaria. The people who are heterozygous dominant, therefore are normal, are more susceptible to malaria. So this relationship between malaria and hemophilia has actually increased the hemophilia recessive allele in the black population, making it a more common genetic disorder than others. Cystic fibrosis is the most common fatal inherited disorder among whites in the United States. What happens with a child with cystic fibrosis is that mucus builds up in the respiratory and digestive tract. It's due to a defective protein that's involved in chloride iron transport in the plasma membranes. And when the mucus builds up, they have breathing problems and digestive problems. These individuals tend to be small and show symptoms of malnourishment. They tend to get serious lung infections, and usually they only survive into their 20s. PKU is phenylketonuria, and it's when there is an abnormal protein that is produced. That protein is phenylalanine hydroxylase, which is the enzyme that converts dietary phenylalanine, which is an amino acid found in animal proteins, into tyrosine. If you don't have this enzyme, excessive phenylalanine builds up and causes brain damage. Newborns are tested in the hospital when they're born. Their urine is tested for this disorder, and if it shows up, the child basically is put on a vegan diet with no animal proteins for the rest of their lives. So they don't consume phenylalanine, and the genetic disorder doesn't express itself. If they did consume phenylalanine, they would die very shortly. Dominant disorders are much less common because people know that they have a dominant allele, and usually if it's a serious disorder, they die before they reproduce, or they don't reproduce because they don't want to pass the disorder that they have onto their child. So some examples of dominant disorders are Huntington's disease. When a person is about 40 years old, they basically go crazy and die. There's a progressive breakdown of brain cells, they lose muscle control and mental function, and it leads to death. One of the problems with Huntington's disease is that people don't know they have it until they're in their 40s, and therefore most people have already had children. They pass the dominant allele onto half of their children. So if a child in their 20s watches their parent die of Huntington's disease, then they come under the realization that there's a 50-50 chance that they're going to suffer the same fate. There is a genetic test, and they can be tested to know if they have it. And one of the reasons that they may want to have that test is because if they do have Huntington's disease, they may not want to have children, so they don't pass it on to them. Achondroplasia dwarfism is the most common type of dwarfism. 99.99% of the human population are homozygous recessive. This is caused by a dominant allele. HYPERCHOLESTEROLEMIA is the presence of one allele causes the person to have half of the normal number of receptors for low density lipoproteins, LDLs. That's the bad type of cholesterol. So if they only have half of the normal number of receptors, then that cholesterol cannot be removed from the blood, or half of it is removed from the blood. If they have two dominant alleles, they have no normal receptors, and all of the bad cholesterol remains in the blood. And that person dies at a very early age of heart disease.